Effective antibiotics are becoming more and more scarce as bacterial resistance to them continues to skyrocket. According to Centers of Disease Control and Prevention, at least 2.8 million people in the United States are infected with antibiotic resistant bacteria each year, and more than 35,000 people die as a result. Being one of the greatest threats to human health and humanity as a whole, antibiotic resistance in bacteria continues to increase, and due to misuse and overuse of antibiotics, this increase continues to grow with each coming day. The misuse of antibiotics often occurs when patients take antibiotics for viral infection rather than a bacterial infection. In short, viruses cannot be harmed by antibiotics in the same way bacteria are due to their distinctly different molecular buildup. Another reason for a dwindling supply of effective antibiotics is that antibiotic research has also ultimately become less profitable for big pharmaceutical companies because of the continual need for developing newer and stronger antibiotics each year. Due to this, most big pharmaceutical companies have abandoned antibiotic research or have severely limited their research into the discovery. This is where Tiny Earth comes in, a network of students just like you and me who are interested or involved in researching antibiotic producers with the hopes of discovering and isolating a new antibiotic. The answer of how this is done is through the sampling of soil from across the United States, with the hopes of one of these soil samples being the home of an antibiotic producing microorganism. Thousands of students sample soil from their backyards and follow a protocol of obtaining and diluting the soil, patching these bacteria, testing them for antibiosis, testing these bacteria samples against other common bacteria, and sequencing their genome. All of these methods and more are used to determine whether the bacteria that might even be lingering in our own backyards can be used to become the future antibiotics that we use to combat the ever-increasing rise in bacterial interventions. At the beginning of my Tiny Earth project, I began by taking a soil sample together with my lab partner from his backyard and bringing the sample into our lab for testing. We begin by documenting our soil with characteristics such as its location, soil type, pH, and other descriptions since the microbiome of soil can vary based on where it was sampled. For example, the soil taken from the arid soil of Arizona will most likely have a distinctly different microbiome than one taken from the humid soil of Florida. Next in our procedure, we dilute the soil sample with water multiple times and spread this diluted soil across an agar plate. We then incubate this plate to grow the bacterial colonies that we had living in our soil to eventually isolate them. The agar plate we use are nutrient-rich dishes used to grow different types of bacteria and other microorganisms, while the incubator is used to provide a warm environment for the bacteria to grow at a faster rate than they would at a room temperature. The incubated plates showcase a variety of growth, but each of the dilutions were useful in narrowing down the amount of bacterial colonies that we had growing on our agar plates, making it easier for us to identify each distinct colony. The next procedure of our project was patch plating. In this procedure, our protocol included sampling each of our now isolated colonies from our agar plate onto a new plate and using a numbered grid to associate each isolated bacterial colony with a number. This new patch plate will become our library, an essential item that will be used for the next part of our experiment. There was just one tiny problem, or more accurately said, one expansive problem that occurred with our previous procedure and which occurred with many others in our lab group. This was the presence of Bacillus mycotes in our samples. Bacillus mycotes love the nutrients of our agar plates and have the tendency to take over an entire dish in the span of hours. This is why when patching our new plates from our dilution plates, we made sure to only sample the colonies that were away from any Bacillus mycotes. Since our group had some samples of interest that were too close to the mycotes, as seen in this picture of our agar plate library, we decided to create a quarantine plate. The quarantine plate effectively involved us sampling the bacteria and streaking it across the clean plate to spread out our bacteria. This is to create a larger sample of our bacteria, making it easier for us to sample it onto our new library agar plate. As you can see, there is still a bit of mycotes, but now we can sample an area of the plate that isn't close to the site of the mycotes onto our new library plate. Next in our project is something known as antibiosis testing. This is where we test our now cataloged bacteria samples against other common bacteria such as E. coli, Staphylococcus, and Bacillus to determine antibiotic production. We first created special plates that were covered with a diluted layer of these bacteria, one plate for each bacteria species, and copied the bacteria from our patch plate onto each of these antibiosis plates. We incubated the samples and came in the next lab period to identify any zones of inhibition that may be present between our bacteria and the bacteria we had spread on our plates. A zone of inhibition is a circular area around an antibiotic-producing bacteria which may affect the bacteria that are around it by inhibiting their growth. Although one of our colonies may show zones of inhibition against one bacteria, it does not mean that we will see this for all bacteria types. This is why we test our samples against multiple different strains of bacteria. For example, a very good antibiotic producing bacteria could show signs of inhibition against E. coli, Bacillus, and Staphylococcus, while an intermediate antibiotic producer could show signs of inhibition against E. coli, but not other species of bacteria. Now back to our own procedure. After identifying the bacteria colonies that had zones of inhibition against the bacteria we spread on the agar plates, we graphed the information in order to visually understand how our bacteria's antibiotic capabilities fared against other known bacteria. At the end of our procedure, we selected 3 samples that showcased both large zones of inhibition and had zones of inhibition against more than one bacteria sample. These 3 samples were labeled as MB-23, MB-24, and CMM-1 based upon our initials to properly catalog them. The next and fourth step of our project was to purify each of our suspected antibiotic-producing bacteria. This is to ensure that none of our supposedly isolated colonies of bacteria have any other species growing alongside with them. We begin by selecting our patches or colonies of interest based on whether they have zones of inhibition against multiple bacteria species and how large these zones of inhibition were. This is how we know if our antibiotic-producing bacteria is a strong antibiotic producer in our scope of testing, and how broad the effectiveness of its produced antibiotic is against the bacteria species we tested in our previous antibiosis procedure. After selecting our patches of interest, we streak these colonies onto a clean agar plate. Streaking involves diluting the bacteria 4 times and spreading each of these dilutions in a zigzag pattern, each streak dilution pertaining to a quadrant of the petri dish with little overlap between each of the quadrants. After streaking, the dilution plate is placed into an incubator for about a day and observed the following day for any additional species being present on the sample, in which another streak plate would be created to further isolate the bacteria. In our own experiment, samples MB-23, MB-24, and CMM-1 were streaked onto new agar plates. Following incubation, we ensured the purity of our samples by verifying that only one type of bacteria was present on our streak plates. The next and final step of our procedure is actually a 3-step process to basically characterize our bacteria samples. We first begin by amplifying the 16S DNA of our bacteria through a process known as the polymerase chain reaction, or known more commonly by its acronym PCR. This process denatures the DNA into single strands, reattaches new primers that specifically bind to the 16S genes, and extends the new DNA, creating millions of copies of our target DNA. Once we have effectively amplified these 16S genes, we are then able to run the PCR sample through a process of gel electrophoresis, which separates the DNA fragments by their size and charge. Once we have our gels fully completed, we are then able to analyze the bands under UV light and compare them to a ladder, which works as the key for interpreting our series of bands. This is how we are able to identify if we have a positive sample, and if our bacteria sample would make a strong candidate for going on to the next step of 16S rRNA sequencing. The step involved us sending our samples to another lab with the equipment necessary to sequence the 16S rRNA of our positive bacteria samples. After we had received our sequencing data, we were able to use a tool from the National Center for Biotechnology Information called BLAST, which stands for Basic Local Alignment Search Tool. Before we could input our sequence data, we would first need to analyze the chromatogram, which, in short, means a visualization of the nucleotides present in our 16S gene of our sample. We would then take our nucleotide sequence and run it through BLAST, working to match our sequence with its nucleotide database and identified organisms with the same nucleotide sequence. In our own experiment, we were able to run a PCR of our samples and run our amplified 16S genes through its gel electrophoresis, but due to what might have been insufficient amplification, 2 out of our 3 gel samples ended up with undistinguishable bands. We were unable to characterize our samples as positive samples, and due to this, we did not end up sending our samples in for 16S rRNA sequencing. Although this was a bummer to see our samples not have a shot at being the next revolutionary antibiotic, it was fascinating to see our samples make it this far. We were able to analyze another group's sequence which had positive bands within their gels and whose samples were sent in for sequencing. We were able to analyze the chromatogram from a program called Finch TV and input the nucleotide sequence to BLAST. The bacteria species that was identified for the G3 group sample was Pseudomonas helminthiensis, with a query cover of 98% and a percent identification of 98.38%, meaning that our sample is a very close match to this genus and species of bacteria. Many times with scientific research, it is a process of trial and error, and not every experiment will conclude with results that are noteworthy or even in our case, end up with results. Looking at the bigger picture, although we did not have viable results at the end of our projects, Many of our peers did have positive samples, leading to the identification of their bacteria samples and furthering the research of discovering a new antibiotic through the Tiny Earth database. As the Tiny Earth protocol is being implemented in classrooms all over the United States, providing students with hands on experience in scientific research and contributing to the global effort to combat antibiotic research, I hope that this video emphasizes the need for antibiotic research and provides a lens into the process of this research through Tiny Earth. Thank you for watching this video, and for more information about Tiny Earth and antibiotic research and the sources that were cited in this video, please visit the description below.